We're going to be jumping into the second part of chapter 8 today. So the second part of cell division, which is meiosis. The second type of division when it comes to cell division is meiosis. Meiosis, however you want to say it. I like to say meiosis because I'm going to show you a little trick on how you remember the difference between meiosis and mitosis. So mitosis, we looked at dividing somatic cells into two genetically identical daughter cells. Well, meiosis is the division of sex cells into four genetically unique haploid daughter cells. So a little bit different. So let's look at these differences really quick. So first of all, sex cells. So our sex cells, those are going to be like the ovaries in your females and your testes in your males. And you create, after this division, the daughter cells are going to be the eggs for the females and the sperm for the males. So that's what we're doing here, is we're creating egg and sperm cells from our sex cells, so the ovaries and the testes. All right, so division of a sex cell, so one cell into four, so instead of two this time we're going to go into four genetically unique. So this time they're not going to be genetically identical, they're going to be genetically unique, which means each one of those four cells are going to be different genetically. And then they're going to be haploid daughter cells. So one thing we haven't talked about is our ploidy number. So our ploidy number is how many chromosomes a cell has. So our somatic cells, they are diploid. So diploid means two copies of each chromosome. And this made sense because we said in our cells we had 23 paired chromosomes, right? 23 paired. And so we're saying we're diploid because we have two copies of each of our chromosomes. That's what that 23 paired is, two copies of each of those chromosomes. So that's diploid. So our sex cells are also diploid, but once we turn them into our eggs and sperm, they then become haploid. All right, so haploid is half the amount of diploid. So half the amount of the chromosomes of diploid. So what we're going to end up seeing, instead of saying 23 paired chromosomes like we saw in our somatic cells when we did mitosis, when we finish up with meiosis, our eggs and our sperm are going to end up having only 23 chromosomes. Right? 23 total. So remember, this is 23 paired, but 46 total. So in other words, we're going to split up the pairs. And by splitting up the pairs, we're going to make it so that each one of our four daughter cells that we create will only have 23 chromosomes, which makes them haploid instead of diploid. All right, so one big question with meiosis is something I always like to ask my class, which is, why sex? Okay, why do why do a lot of organisms, living organisms, participate in sex, or what we like to call sexual reproduction? So when we think of like smaller organisms like bacteria and whatnot, they do asexual reproduction, which means they can just reproduce on their own. they can just split off and continue to reproduce on their own, or they can bud on their own. We have asexual plants that can do that. There's actually even asexual lizards and other type of fish that are asexual, where they don't need another type of individual in their population to be able to produce offspring. But what we see a lot in animals, specifically like over 99% of animals participate in sexual reproduction. And we look at plants, they also reproduce majority through sexual reproduction, somewhere probably around 85+%. And then fungi, they do both. They do asexual and sexual reproduction. And so sexual reproduction is very popular in life. And so why sex? Why do these living organisms participate in sexual reproduction? And the main reason for this is because during sex, we are going to mix up the DNA of the father and the mother to create the offspring. Where in asexual, you're just saying, all of my DNA is going to my offspring. So you're pretty much making an exact copy of yourself every time you do asexual reproduction. Where sexual reproduction, we're saying our offspring are going to be a mixture of the genes from the mom and the genes from the dad, and then that's going to be the offspring. And so in life, this can be very beneficial because a lot of the times as the environment changes or as things happen, so let's give a good example of COVID-19. So this is a novel virus that's affecting a lot of people. Well, there might be some people that are actually immune to COVID-19 because they have certain genetics makeup in their genome. And so let's say COVID-19 was even more deadly than it is right now, and it was wiping out a whole lot of the population instead of roughly 2%, 1 to 2% that get infected. Then only those that would be immune to it would probably live on and move on. And so during sexual reproduction, we mix up the genes of the human race, or which other population, whether animal or plant that's doing it, we mix up those genes inside so that we spread genes out across the whole population so that there are better chances for people to be immune or to be evolutionary more advanced to be able to go on and procreate themselves. And so that's why a lot of living organisms utilize sexual reproduction. All right, and so for us as humans specifically, and most animals that do sexual reproduction, we have to have our sex cells go through meiosis to create our gametes. So our egg and our sperm, these are also called gametes. All right, to be able to create gametes, and then what we do is we have the egg and the sperm come together. So both of them are 23. So you're gonna have, let's say here's our sperm and there's our egg. Both are going to be 23 chromosomes, and when they come together to make one, it's a bad arrow. When they come together to make one, so when they fertilize to make one new offspring, then it now has 23 paired, which is a total of 46 chromosomes. And that's how we get back to being a diploid organism, a diploid somatic cell that then divides and becomes a fetus, and then an infant, and then eventually an adult type of thing, right? So this is a very important part of sexual reproduction, is to break down our cells to only having 23 chromosomes each, so that they can then come together to fertilize to create a new offspring. Otherwise, if we were doing diploid, we would be sending 23 paired each, and then that next offspring would then have 46 paired, and then 92 total, and then the next offspring would have double that and double. So we want all of our offspring to have the same amount of chromosomes moving forward. So that's why we do meiosis, to break our chromosomes into packs of 4 with 23 each, and then those 23 will come together from 2 different individuals to create our fertilization point to create our new offspring. All right, so now we're going to look specifically at the different steps of meiosis. And what we're going to see in meiosis, actually before we do that, let's talk about something called crossing over. So crossing over is another way in which we mix up our genetics so that our offspring are even more unique than they could be if we just did it regularly through this process. And so what happens is in each one of our sex cells here, so our eggs and our sperm, not eggs and our sperm, sorry, our ovaries and our testes, in each one of them, before they go into, or the very beginning part of meiosis, is they do something called crossing over. So if we remember, in our cell cycle, we get to a point where we create all of our sister chromosomes. So we make an exact copy of all of our chromosomes. So let's say this is chromosome 1 from mom, and we have our exact copy here, right? That is chromosome 1 from mom. And let's say this is chromosome 1 from dad. So we've made our exact copy, once again, chromosome 1 from dad. And if I remember right in our thing we talked about last time, that mom had brown hair and brown eyes. So we're going to put those codes in here. And dad had blonde hair and blue eyes. So what happens in crossing over is in our cells, so our testes and ovaries, depending if you're male or female, all of our sister chromosomes are going to line up right next to each other with their homologous chromosome, right? So homologous are the same chromosomes with the same genes, and then sister chromosomes are the exact same copies. And they're going to line up together, and what they're going to do is they're actually going to switch their DNA, their genes, amongst their homologous sister chromosomes. So what you could say is, okay, let's say we're going to take out this part here and replace it with this part here. Okay, so we had crossing over take place. So we mixed up this chromosome together, and then we can say, all right, we're also going to switch this part. And so what happens in all of our 23 sister chromosome, paired sister chromosomes, our total of 46 sister chromosomes, are all going to line up with each other on their homologous chromosomes, and they're going to do this switching, crossing over, where they're going to be switching different genetic material with one another. And what this does is this guarantees that our sex cells that get divided into our gametes, so to create our egg and sperm cells, our 4 daughter cells, that they are going to be genetically unique. There's no way they're not going to be, because we just mixed up all of our genetic material with each other. And so we're going to make very genetically different cells at the end of this. And once again, the reason why we do want to do that is because we're going to engage in sexual reproduction, and sexual reproduction allows us to take our mix-up genetics and with somebody else that has mixed-up genetics, put our two gametes together, the sperm and the egg, and then those mixed-up genetics will then combine together to create a unique genetic set for that individual. That's this whole process. All right, so now we're going to actually look at meiosis and see how it works. And meiosis is going to be very similar to mitosis, especially you're going to see, but because we're going to make 4 cells, we actually have to go through the whole process twice. So we're going to see prophase, metaphase, anaphase, telophase, but they're all going to be one of it. So we have prophase 1, metaphase 1, anaphase 1, telophase 1. Then we're going to do prophase 2, metaphase 2, anaphase 2, and then telophase 2. And at the end of that, we will end up with these 4 cells because we went through this whole process twice. Where we saw it when we did mitosis, we went through it once, we ended up with 2 cells. Because we're going to do it twice, we're going to end up with 4 cells. So you see here are 2 phases of meiosis. So we first have prophase 1, metaphase 1, anaphase 1, telophase 1. Then we're going to go through it again and do prophase 2, metaphase 2, anaphase 2, and telophase 2. And you'll see that there are a lot of similarities to mitosis, but there's going to be some difference. So the first part we're going to see here is very similar in prophase 1, where we're going to see the breakdown of the nuclear envelope. We're going to see chromatin, which is lightly packed DNA, go to chromosome, which is tightly packed DNA. And then we're also going to see our centrosomes migrate to opposite ends of the cell. And the biggest thing that happens here, so we just talked about crossing over, is crossing over also happens here. So that process of crossing over happens here in prophase 1. And so when I draw our chromosomes here, I showed them as kind of mixed up DNA with the reds and the blues, kind of like we did with our crossing over our homologous chromosomes, just to represent that in prophase 1, crossing over takes place. So that's the main difference between prophase and mitosis and prophase 1 in meiosis, is that crossing over takes place and we mix up all that DNA so that we can create genetically unique daughter cells. Other than that, the other things happen just the same. Metaphase 1, so the difference we're going to see here. So before we had our sister chromosomes line up in the middle. This time we're having our homologous chromosomes line up in the middle. And I know I haven't drawn them here, but realize that they're all kind of mixed up. So this would be one from mom and one from dad, 2 from mom, 2 from dad, yada, yada, yada. All 23 of them, right? Because we have 23 paired chromosomes, our homologous chromosomes, are all going to be lined up. And then our centrosomes are going to attach those protein spindles once again now to our homologous chromosomes. So that's metaphase 1. Anaphase 1, we're going to see that we're going to split our homologous chromosomes from each other. So in metaphase, in mitosis, we split the sister chromosomes, so we split these in half. This time we're just splitting the homologous chromosomes. Okay, so split apart our homologous chromosomes. Then in telophase, it's going to look very similar. So in telophase, we're going to build back up our nuclear envelope. Our our chromosomes are now going to go back to chromatin, and then we're going to pinch off. So cytokinesis is going to happen. Which is it's going to split the cell in two. And so at the end of our first round of meiosis, we're going to end up now with 2 cells. All right? Now, how many chromosomes will be in each one of these 2 cells? So what we did here, remember, we went our sex cells, so our testes or our eggs went through the cell cycle, so they made a copy of all their cells. So starting here, we had 46 sister chromosomes, which was a total of 92 total chromosomes. So at the end here, we ended up splitting all that so that we end up now with 23 sister chromosomes, which is a total of 46, and 23 sister chromosomes, once again, which is a total of 46. So remember, we're trying to get to 4 daughter cells that only have 23 chromosomes in them. Right now, we have 2 daughter cells that have 46 chromosomes in them, and we just call those 23 sister chromosomes. So what we need to do is have each one of these cells come and go through prophase 2, metaphase 2, anaphase 2, telophase 2, so that we can split their sister chromosomes and then end up with 4 cells instead of 2. All right, so let's look at prophase 2. So prophase 2, once again, our nuclear envelope starts to break up. Our chromatin goes back to chromosome, back to chromosomes, so we can start to see them here. Our centrosomes migrate to opposite ends of the cell. So just like we saw in mitosis. Then in our metaphase 2, our sister chromosomes line up in the middle again. And our centromeres attach spindles now to the sister chromosomes. So metaphase 2. Then in anaphase 2, we split our sister chromosomes in half and we tear them apart. So we separate those sister chromosomes that were all lined up here in the middle. And then telophase, we reverse everything that happened in prophase. So we have our chromosomes go back to chromatin. We start to build back up our nuclear envelope. So the nucleus. And then we have cytokinesis happen and split our cell into two. So we take one of these, goes through that creates two cells. We take the other one, have it go through that, and that creates two more cells. So if we started with one of these cells that had 23 sister chromosomes, and we split those sister chromosomes in half, put them on the other end of the cells, pinched off, we then end up with a total of 23 chromosomes here, 23 chromosomes here, 23 chromosomes here, 23 chromosomes here. So we now have been able to create 4 genetically unique haploid daughter cells from our one original sex cell. So if this was a female, then we would have created eggs. If this was a male, then we would have created sperm. So that is meiosis. That is how we're able to create these sex cells that then allows us to engage in sexual reproduction to create offspring that are genetically unique because they're a mesh up of not only the mom, but the dad and all their genes meshed up together through that fertilization process. Now, there's an interesting thing that happens. What happens if there is a problem in meiosis? And usually that problem would happen in anaphase. So let's say in a situation where there's a problem, and this sister chromosome didn't split in anaphase. All the rest of them did, but this one didn't. And let's say this is chromosome number 21. All right, so then as it splits here, what we end up happening in one of our cells, we're going to end up with 22 chromosomes, and the other cell, we're going to end up with 24 chromosomes. All right, so let's say these are egg cells, and we have a sperm cell. So here's our sperm cell that has a regular 23. Well, if this one and this one combine together to create offspring, they would then have not 46 total, but 47. And specifically, if this is chromosome 21, that means chromosome 21 would have 3 chromosomes. And this is a genetic disorder we call trisomy 21, which means 3 chromosomes for chromosome 21. And what this actually does, this actually has a genetic disease that's associated with it, which is called Down syndrome. So offspring that have Down syndrome, what happened is that there was a problem during anaphase 2, and the chromosome didn't split like it was supposed to, the sister chromosome, and eventually ends up that individual now has 47 paired chromosomes, with that extra chromosome being chromosome 21. Now, if this happens in other chromosomes, most likely what happens is that you won't get fertilization, or the egg will abort. It won't go to full term. But there, like I said, in the case of Down syndrome, it does go to full term, and that's why you have people that are Down syndrome. There's other types of genetic disease that happen like this, but they involve the sex chromosomes. So one thing we haven't talked about yet is our sex chromosomes. So to show this really quick, I'm gonna erase telophase here. So our 23rd chromosomes are our sex chromosomes. And if you are a male, you have an XY chromosome. And if you're female, you have XX chromosome, which means you have the same homologous chromosomes. You're the same on your sex chromosomes. But if you're male, you have one that's X, and then you have the smaller one that's Y. And there's different genes on our X chromosomes and our Y chromosomes that allow us to develop whether in male or female. So there's specific genes that help with production of testosterone and whatnot in our Y chromosome that females don't have. And there's genetics in there for like estrogen in our X chromosome. So you're saying, well, men have X chromosome, but because they only have one pair, it's not as produced as it is in our females. Well, when we're doing anaphase 2, there are also times where you could have a sex chromosomes that don't split, right? And so sometimes you can end up with an individual that has 3 X chromosomes. And people who have 3 X chromosomes, usually that's not a problem and they can still go to full term. But then there's people who will end up with 2 X's and a Y. And these are individuals that have a disease called Klinefelter's. And Klinefelter's is where you have, like I said, this XXY, and people who have this disease often are physically female, and they tend to be shorter. They tend to have what we call a web neck. They have a really short, kind of wide neck. And then there's even times where people can be what we call X naught. So that means they have 1 X chromosome and they only have 1 sex chromosome. And so there's different things that can happen, and this is how sometimes you see different things with people that are intersex and whatnot, because they have multiple sex chromosomes and they have that issue. So they don't really develop specifically just as a male or specifically as a female, just based off something wrong happening in anaphase 2 where that doesn't quite split like it's supposed to. All right, so that is meiosis. So meiosis and mitosis, because they sound so much similar, we got to figure out ways to kind of separate them in our mind. And so the way I always do it is mitosis. So mito is a somatic cell, and so it goes through mitosis. And then I say meiosis. Meiosis is to make a mini-me, right? So if I want to do sexual reproduction and create offspring and make a mini-me, then my cells have to go through meiosis. So that's how I always, when I was in college, figured out the difference between mitosis and meiosis, so I know which one is which. All right, that is the end of chapter 8.